Susan, I wanted to ask you a question around implementation at Sonnet from a process and equipment perspective. If you could kind of walk us through what the chronology of the project looked like at Sonnet, that would be great.
Sure. So we started in fed batch mode and found that a 15-day fed batch process, we were losing so much drug from day 10 onwards because of proteolytic clipping that we thought, okay, we'll keep it a 10-day process. But when you keep it a 10-day process, you start, the productivity levels are drastically reduced. So as a combination effort in parallel with making doing the fed batch development, we also initiated a intensified perfusion process. And really, it's the same bioreactor at a smaller scale. So we were actually getting as much drug as we need, because you have to remember cytokines are delivered in nanogram per kilogram of body weight amounts. So my doses are in this, in the low double-digit micrograms per patient. 70-kilo patient, you're looking at 20 to 30 micrograms, whereas antibodies are, multiple, even hundreds of milligrams. So the relative scale I needed to be at and the relative productivity I needed sets the bar on the low side, which is very valuable in this case. So we went to perfusion because the bioreactor was virtually the same. We started at 50-liter fed batch. We went to 20-liter perfusion. And we got almost 10 times as much drug from that same 15-day 20-liter perfusion as we got actually more than 10 times the level of output than we did from a 50-liter fed batch process. Mm-hmm. So that's kind of what kicked us off. The bioreactor is the same. The equipment is virtually the same. There's an addition of, in our case, an ATF system, which is a big filter system that keeps the cells, allows you to sip off the drug. And we're doing kind of a sipping of drug on a 24-hour volume replacement basis. So for the first half of the process, we take out, one volume per day, and we move to 1.5 volumes per day, and then 2 volumes per day for the last few days. We are maintaining a 15-day process because we don't need grams of the material. We actually, we get grams, and that's as much as we'll need. So we don't extend it beyond that 15-day time threshold for the end of the process, which also adds a regulatory simplicity to it, that it's not extended continuous manufacturing for months on end. So it's a short-term 15-day process.
Great.
Did I answer the question?
I think so. Yeah. And as you've been speaking, Susan, we've received a few questions. So before we get to those poll results, I'm just going to ad-lib a little bit here, and we're going to address. There's a few questions here that deal with perfusion. People looking for a little bit more clarity on that. Miles, I'm asking my assistant here in the office, Miles, my producer, if he can expand that Q&A box so that I can see those full questions. Is up? Oh, there we go. Yeah. Give me question 2. So here we go. I really consider the perfusion cultivation advantages, but can we really run it for 50 days without experience? For example, membrane blocking. Do we have nowadays equipment that can hold the process in a stable way? So listen, if you want clarification on some of these questions, we may have to go back to the asker, Susan. But if you can make a little bit of sense out of these questions and maybe address them, that'd be terrific.
Sure. So it's not 50 days, it's 15. So I'm running a 15-day, not 50-day process for that's the first thing. And the second thing is one of the challenges that you have with biomanufacturing period is foaming. But it becomes exacerbated when you have a filter system like an ATF to add any kind of anti-foam to the manufacturing process, because it does coat the ATF. So you have to be mindful of foaming in a perfusion process, and we've been successful at minimizing the foaming without having to add anti-foam, but it can cause a film to exacerbate the ATF system and plug it up in essence. So you have to, there are some process modifications that you have to be mindful of.
All right. Yeah. Miles, give us that last one, if you would. Does intensified perfusion alter product quality since cell culture lasts many weeks?
Parrish can probably answer this as well, but what we've seen is very consistent processing over time in terms of product quality. But again, we're not extending our perfusion system to be months. We're maintaining a 2-week manufacturing scale process. So we have not seen any issues associated with that.
Okay. Parrish, did you want to weigh in on that one?
Well, fouling of ATF and other filtration systems for cell recycle is commonly reported. And to Susan's point, antifoams can exacerbate the fouling of filtration systems. And when they get fouled, they start changing the molecules that they allow through. So they can actually modify the product quality as a result. So it's important to have control over your foaming or to try to reduce it as much as possible, therefore minimizing the amount of antifoam that you require. And if the filter plugs, you need a backup filtration system on the bioreactor to switch over to get back to the filtration performance that you started out with.
Excellent. So, Miles, the question from Olivia addresses a topic that I wanted to get into anyway, so let's take that one. How did you qualify technology and equipment needed for perfusion intensification? And Susan, I know I know at least partially what your response is going to be to my follow-on to that question, but around the qualification of the technology and equipment, I also want to talk about the investment considerations. So why don't you tackle that question? What, you know, from your perspective as the as the biotech and not the CDMO, did you have any influence or commentary around the qualification of technology?
Yeah, well, as it turned out, again, because the platform, I was very familiar with it. It was based on Sartorius technology to a very large extent, for which I am very familiar. Worked with it before at previous CDMOs, and because I wasn't making a lot of changes from what I already knew, I didn't have a lot of concerns about qualification, because we're still, you qualify it the same way you would any system for biologics. And since I'm a virtual company, I outsource everything. And so we evaluated the quality systems, the qualification systems, the validation master plan of the CDMO we've chosen, and their process for qualification of equipment was adequate and thoughtful, and it covered all the risk assessments that we had considered as important to us from the point of view of the product quality coming out. I've seen a lot of data from this particular company's internal pipeline as well, and it's very consistent. They maintain a high degree of control, which tells you that the qualification is aligned with the ability to control in a tight manner the manufacturing process. And by the way, we are preclinical. We're just about to go into the clinic. So we're talking about very early manufacturing stages here, and we're still getting very consistent results from the manufacturing process we're running.
Excellent. And from an expense standpoint, there was no direct expense incurred to you in the relationship with the CDMO?
No, I did not have to buy any unique equipment. In fact, it's kind of against my principle. If you go to a CDMO, I think sticking with as much standard stuff that they know how to run, know how to use, and are good at is a good idea. Yeah. So I let them drive the bus from that perspective, making sure that whatever technologies, viral filters, or things that were that change product quality were evaluated so that we weren't biased towards one vendor or another, but actually did what was best for the molecule when it came to the rest of the parameters that needed to be monitored for product quality attributes in critical process parameters.
Okay, Parrish, I want to turn to you for the internal manufacturer perspective. So if I'm my own manufacturer, two-part question. One, what qualities should I be looking for? How should I qualify perfusion technology per the audience question? And two, what should my expectations around investment be?
Regarding qualification, like any other manufacturing system, you do need to qualify the cell perfusion system. So you go through a normal installation and operational qualification exercise and validation protocol to do that. It's very straightforward. And then for performance qualification, you do a production run using real cells and real culture to demonstrate the cell separation efficiency of the perfusion system. And in doing that, you can also validate the product quality that's produced by that system. With regard to expense, there are many folks that think that going to perfusion is going to reduce investment. And in fact, that's not been reported in the literature. You might shrink the bioreactor by a factor of 10-fold in scale, but that does not shrink the capital cost by 10-fold because you have to add sterile media supply vessels to support feeding the bioreactor. And on the backside, you have to add sterile vessels to collect the sterile filtrate coming off of the perfusion system. So in fact, you're adding a number of pieces of sterile processing equipment to the footprint of the facility to in fact go to implement process intensification using perfusion. So just to add to that, a number of companies have evaluated the cost of goods impact of doing perfusion. And again, what's been reported is that there's no reduction in cost of goods. And in fact, because of the cost of media feeding the bioreactor, the cost of goods can in fact be higher when you're doing process intensification.